Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. Sexual reproduction in flowering plants with structure of seed, seed dormancy and its significance. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain the structure of seed along with its significance. Seed is a characteristic feature of spermatophytes like angiosperms and gymnosperms. In angiosperms, after completion of fertilization, the fertilized egg cell it becomes into embryo. At this time, the entire ovary it becomes into fruit and ovules present inside the ovary it becomes into seed during sexual reproduction. In angiosperms, seed is the final product of sexual reproduction. Generally, the seed is defined as a matured integumented megasporangium generally. But morphologically, the ripened ovule is called seed and ripened ovary is called fruit. In the structure of a typical matured seed, three parts are present. In this, the first part is seed coat, the second part is embryo, and the third part is cotyledons. Seed coat, it is the outer protective covering of a seed. Now it is developed from integuments of ovules. This seed coat, it helps to protect the embryo from injuries and along with that it prevent drying out. The seed which is developed from bitegmic ovule. In such seeds, the seed coat contain two layers. The outer thick hard leathery layer which is developed from outer integument, it is called testa, and the inner layer it is a thin and papery membranous in nature, it is developed from inner integument, it is called tegmen. But in some of the seeds which are developed from unitegmic ovules, these seeds it contains only one layer in seed coat. The seed generally it attaches to the pericarp wall or fruit wall with the help of a stalk, it is called funiculus. In this, the ovule or seed it attaches to pericarp or fruit wall with the help of a junction, it is called hilum. In the structure of a seed coat, a minute pore is present, it is called seed pore. The second part is embryo. Embryo is the most important part in the structure of a seed. Generally, this embryo it is developed from fertilized egg cell. In the structure of a fruit or seed, now the embryo it is packed with the help of a temporary food supplying tissue, it is called endosperm. During seed maturation, this endosperm it may be present or absent generally. The third part in the structure of a seed, it is cotyledon. An embryonic leaf in seed-bearing plants with one or two or more, which are first leaves to appear from a germinating seed, these are called cotyledons. These cotyledons are generally swollen structures due to storage of food materials, and these food materials are supplied to developing seedling at the time of seed germination. Based on the number of cotyledons present in the structure of seed. The seeds are two types. In this, the first type is monocotyledon seed and second type is dicotyledon seed. In the structure of monocotyledon seed, by taking maize as a example, a maize grain, it is a scientific name is maize. It is a dry, one-seeded, indehiscent fruit of caryopsis. In the structure of these grain, both pericarp, it is called fruit wall, and seed coat, both are fused to each other. It is not possible to separate. This covering of grain, it encloses two major parts. In this, one is endosperm and another one is embryo. The major portion of grain is occupied by a large starchy endosperm, and this endosperm generally it is surrounded by a single celled thick layer, it is called aileron layer. In this aileron layer, the cells are filled with starch and proteinaceous grains. The embryo, it consists an upper shield-shaped structure. It is called scutellum. Now, the epidermal tissue of this scutellum laying and it is a direct contact with endosperm tissue. This epidermal tissue, it is generally secretory in function along with absorption function. These cells, it secretes some of the hormones like gibberellins into endospermic tissue and these enzymes like alpha amylase. These enzymes, these are responsible for breakdown of stored nutrients and these nutrients are converted into soluble state. These soluble nutrients, these are absorbed by epithelial cell of scutellum and these are transferred to a growing embryo. The scutellum, it is attached to lateral side of an embryonal axis and in this embryonal axis at one of the end it having a structure, it is called radicle. This radicle, it is covered with a protective sheath, it is called coleorhiza. The another end of embryonal axis, it contains a plumule. This plumule, it is also surrounded by a conical protective sheath, it is called coleoptile. In the structure of dicotyledon seed, by taking bean seed as an example, in the structure of a bean seed, it having a concave side and it is slightly dark in nature and possesses a white elongated scare, it is called hilum. This hilum, it represents the point where the seed attaches to funicle. A minute pore, it is present just adjacent to hilum, it is called seed pore. This seed pore, it performs absorption of water during seed germination. There is a short ridge on outer side of this hilum, it is called raphe. In the structure of bean seed, the seed coat having two layers. In this, the outer layer is called testa and it is hard, thick and smooth in nature with different colors. And this testa, it is developed from outer integument of ovule. The inner layer, it is membranous in nature and thin, it is called tegmen. It is difficult to separate both testa and tegmen in the structure of seed coat. Each coat, it encloses an embryo with two large white and kidney-shaped cotyledons. The cotyledons are with reserve food material in the form of carbohydrates or proteins. The microfile end of embryonal axis, It is a rod shape and slightly plugged. It is called radicle. The outer side of embryonal axis lie in between two cotyledon, it having plumule. The portion of embryonal axis between plumule and cotyledon node, it is called epicotyle, and the portion of embryonal axis present in between radicle and quaternary node, it is called hypocotyle. The significance of seed. Seeds are the end product of sexual reproduction, and it generates new genetic combination that helps to express variation in next generation. The seed formation is the very most important step in plant for plant dispersal because the seed possesses a better adaptive strategy for dispersal into a new habitat and also helps in colonization of the species in new area. It helps in germination of new plant. The cotyledons which are present in the structure of a seed, these cotyledons, it nourishes young seedling up to when it performs photosynthesis. The seeds are the main basis in agriculture. With the help of dehydration and dormancy of matured seed, it is a crucial step to store the seeds throughout the year and also to produce next generation. The hard seed coat, which is present around the embryo, it protects the embryo from various factors. That means the seed coat is protective in nature. The next topic is seed dormancy. The time which is present in between maturation and germination of seed, generally it is known as seed dormancy or dormancy period. The presence of dormancy in seed is the most important characteristic feature because this character it helps to store the seed for many years with viability. The state of inhibited germination, it may take place with the help of either external factor or internal factor. If the seed germination it is inhibited by internal factor, such condition is called dormancy. If the seed germination it is inhibited by external factor, that phenomenon is called quiescence. This seed dormancy it is considerable advantage to plants. It helps to sustain in various adverse and environmental conditions generally. There are three important reasons for seed dormancy. In this, the first reason is impermeability of seed coat. If the seed coat is very thick and hard in nature, such type of hard seed coat, it prevents the entry of water into the seed. Because of this impermeable nature of seed coat, it may lead to seed dormancy in most of the seeds. The second reason is dormant embryo. In the structure of a seed, if immature embryo or dormant embryo is present, that also causes seed dormancy. The third reason is germination inhibitors. Some of the inhibitors they can inhibit the growth of seedling at initial stage. In such condition also seed dormancy may take place. Seed viability. In some of the species, the viability it may be for hundreds of years. For example, Lupinus arcticus plant. The seeds of this plant these are excavated from Arctic tundra. The seeds are germinated and produced flowers after an estimated record nearly 10,000 years of dormancy. These are the Lupinus arcticus seeds excavated from Arctic tundra. In some of the angiosperms, seeds are viable only for few months, but in most of the species, seeds are viable for years. The oldest matured seed that has grown into a viable plant, that is date palm. The scientific name is Phoenix dactylifera. The seeds about 2000 years old. These are recovered from excavation at King Herod the Great Palace on Masada in Israel. It had been preserved in cool and dry places, not by freezing. Such type of oldest seeds with 2000 years of dormancy, these are germinated in 2005. Now it becomes into this date palm plant. The lotus seeds are viable for 1000 years because the lotus can age for 1000 years and even survives freezing weather. Because of its genetic makeup can combat stress, starchy and proteins in lotus seeds, they remain helpful to maintain the seed viability up to 1000 years. These are the seeds of lotus. Some of the methods it helps to overcome or break the seed dormancy in some seeds. The first method is scarification. In this scarification, the seeds which have hard seed coat, such type of seeds, these are roughed on rough surface to make thick seed coat into thin seed coat. Sometimes such type of a thick seed coat can be removed in this method to make the entry of water, oxygen into the seed for germination. The second method is stratification. In this stratification method, the seeds are kept at a low temperature and in the presence of oxygen and water for some time so that the embryo can complete its germination period and it promotes seed germination. It is the method called stratification. In next method, light requirement. In some of the plants, the seeds required light for seed germination. Such seeds are called photoblastic seeds. The dormancy of these photoblastic seeds can be breakdown by treating the seeds with red color light with various wavelength. The last method is viability. Viability is called existence of life in a seed. This viability can be tested with help of a test. In this, the viability of seed can be found out by 2,3,5 triphenyl tetraazoleum chloride. By keeping the seeds in this TTC solution, the embryo it will be converted into pink color that can indicates its viability. Finally, the seed dormancy it can break down with the help of gibberellins or gibberellic acid as a hormone. The next topic is seed germination. The process by which the dormant embryo wake up and it begins to grow, such phenomenon is called seed germination. The embryo of seed it grows into a sporophytic plant. In this, the radicle it can produces primary roots and plumule it can develop into a shoot and lateral structures in plant. So finally the cotyledons it will be degenerates. During seed germination, we observe 5 important steps or changes. In this, the first one is imbibition. The second one is respiration. The third change is effect of light on seed germination. And fourth one is mobilization of reserves during seed germination and role of growth regulators. And the final step is development of embryo axis into a seedling. With the help of these 5 steps and 5 changes, the seed germination takes place. Based on the nature of seed germination and behavior of cotyledon during seed germination, this germination process is 2 types. In this, the first type is apical germination and second type is hypoaxial germination. In apical germination, due to faster hypocotyl growth and elongation than epicotyl in the structure of a cotyledon, it push the cotyledons to outside of the soil. Now such type of germination is called apical germination. In this, the cotyledons are pushed out to the outside of the soil. Example for apical germination like cotton, papaya, castor, onion, cucurbits, tamarind, French bean and mustard. In second type, it is hypoaxial germination. Due to faster epicotyl elongation than hypocotyl, the plumule it come out from the ground and cotyledons remain present under the soil. Such type of germination is called hypoaxial germination. This hypoaxial germination it is generally seen in maize, rice, wheat, coconut, gram seeds, pea seeds, peanut and mango plants. One of the special feature it is observed in few plants like Rhizophora and Avicennia. It is called vivipary. So vivipary it is a special type of seed germination. In such seeds, there is no resting period in embryo because after embryo development, immediately a seed germination takes place inside the fruit. Still, the seeds are present in the structure of fruit and fruit is attached to mother plant. Now, such type of seed germination, it is called in situ germination. It is vivipary phenomenon. This vivipary phenomenon it is generally observed in Rhizopus and Avicennia. Some of the factors they affects the process of seed germination. In this, the seed germination takes place in the presence of favorable conditions like temperature, moisture and air. Any minute changes in the environment it have harmful effect on growing embryo, so that favorable environments are required for seed germination process. The first factor is moisture. The cell of embryo generally it contains 10 to 15% of water even in dormancy period. For the activation of seed germination, the seed it must require 75 to 90% of water in the structure of seed. The second factor is oxygen. The embryo it required energy during seed germination. Such energy it is released from oxidation of organic substances with the help of respiration process. These respiration process require oxygen for oxidation process. The next factor is temperature. The suitable temperature is essential for germinating seed, especially seeds are not germinated in the presence of 0 to 5 degrees centigrade and above 40 degrees centigrade. The favorable temperature for seed germination is 20 to 25 degrees centigrade temperature. It is a suitable temperature for seed germination in majority of plants. Food and nutrition. The growing embryo it requires nutrition during seed germination process. In this, the embryo generally it depends upon a storage food material. It may be present in either cotyledon or it may be present in endosperm of germinating seed. Light is the most important factor. In most of the plants, they do not require any light. for the formation of first leaf. Adversely, the rate of germination is very fast in the absence of light, but for some plants, light is very essential factor for seed germination. Certain epiphytic plants like orchid seeds. These orchid seeds, these are very minute in structure. These are considered as world's smallest seeds. In the same way, thousands of thin seeds are present in ficus and some of the parasitic species like Orobanche seeds and Striga seeds. Very most important thing, the largest seeds in the world, it is seen in Coco de Mer, the seeds of palm tree. The scientific name is Lodesia maldivica. It is called double coconut. It can be reached about 20 inches or 30 centimeters long and nearly 40 pounds, that is 18 kgs in weight. So that such type of Lodesia maldivica seeds are considered as world largest seeds. Thank you for watching. If you like this video, subscribe my channel, like and share my videos to your students and friends.